tropics, belong to the tropical seasonal forests, are mainly growing in Hainan and southern Yunnan. But the day after tomorrow, you will going to Hainan. So today I will primarily talking about the Hainan. And in Hainan, actually, if you see this map, we have the two big of the long-term ecological research network. One is we call the CERN. CERN actually operated by the Chinese Academy of Sciences, and the CFERN and operated by the Ministry of Forestry now. They call the Forestry Grassland Administration. And along this side, the eastern part of the China, they have full distributed the forest. And in Hainan province, actually we have one of the station we call the Jianfengling, and tomorrow you will actually visit there. On another side, still situated in the tropical is the Xishuangbanna. This side is operated by the Chinese Academy of Sciences, and the Jianfengling is operated by us. So tomorrow, my colleague and I will accompany you to visit there. And this side actually started the observation is a long time ago, nearly 50 years, and then officially established this station in 1986. So actually, this is we have a different level. I have the provincial level, ministerial level, and then the national state level. The state level means the Jianfengling belong to this category. And I don't mention too many sides. And here is the Jianfengling and the Bawangling, and then Diaoleshan, Wuzhishan. All these, we have some work in there because it's a large area anyway. And this is a map showing how we, establish a monitoring system and covering the different scale. The largest scale we call the 60 hectare primary montane rainforest. And you will see that picture, actually Stephen Hubble and visited there. More than 20 years ago, actually, I took the delegation with the Canadian, oh, sorry. Okay. Anyway, just short my speech on the largest dynamic monitoring pole established in the Jianfengling. Even up to now, based on my understanding, it is the still largest dynamic monitoring plot, especially in the tropical forest. And we follow the protocols set by the CTFS and the Smithsonian Institute, as the Richard know everywhere. No, and later on we also joined the Chinese Forestry Biodiversity Network, monitoring network. Actually, this network is managed by the Chinese Academy of Forestry, particularly my colleague Mark Keping, and done lots of work coordinating the research. Here, not just the one of the setting up in the primary mountain rainforest, another one set up in the 2021, 62, in the secondary regenerated rainfall forest. And then plus so many satellite, 1 square meter grid satellite plant around in this area. We try to have a big scale understanding how the vegetation, the spatial pattern and the biodiversity issues. The tropical forest actually, as you know, is very rich in biodiversity. Here I give you some of the figure. In Hainan Island, and the tropical forest actually hosts 4,000 species, vascular plant, accounting for 18.4 of the total species in China. and 421 species of bird and beast, relatively 26 and 21 of Chinese bird species. And Xisong Banna is very similar, rich in biodiversity, but I wouldn't detail about that. And this is the slide showing some of the figure of what we had in Hainan. One is particularly in Jianfengling area, is various species hosted by the rainforest, 31 to 38 of the Hainan province. So it's very rich in terms of biodiversity. And then the woody plant growing in the Jianfengling is reach 250 species per hectare. So within this 60 large monitoring sector plot, we will have the largest number of the individual 485,000 individual species which we counted. So you can understand it's pretty rich in biodiversity. Now I talk about the disturbance impact. Actually, we couldn't figure out exactly what's the historical the forest coverage, but based on the literature, we can know probably more than 50 of the percentage of the forest coverage in this area, in this Hainan Island. But due to the war and the overlogging and then clear up for agricultural uses, so the tropical forests gradually reduce. And this map you will see, in 1950, at that time is tropical forest fully covered the whole island. And then in 1958, you will see lots of forests clear up and then, overlogging and, spatially distributed in this island. Again, also in 1966, 93, you will see continuously decreasing, but after 1998, since China initiated the natural forest protection program, and then all the natural forest is put into protection. And you will see this is the old growth here, and then secondary forest, and then lots of plantation. I give the one example, the rubber plantation. You will see the whole area along this island, lots of plantation, monoculture. In addition to rubber, and also have the eucalyptus, and we introduced from Australia, and they grow very fast anyway. And right now you will see, in 1986, again, the old growth, you will see dark green, and then secondary and the rubber. The rubber right now is still increasing because they have very good economic value. So in the very recent, nearly 20 years, we calculated those land use, land cover changes. You will see construction land and still increasing, even though slightly, slowly. But that's resulted from the conversion of forest land, cropland, but even though the natural forest protection is an initiative. And this is the current figure. The current figure, the whole island is forest cover is 62. It's very much increasing, but it's really contributed by the increasing monoculture plantation rather than we remaining the natural forest. So this is a basic facts. And this slide showing, when we have the slash burning and over harvested, you will see the site like that. And then over the time, the natural regeneration, and then the different phases through the ecological succession, and eventually this is a primary old growth. You will see this. But the question that can we have our degraded natural forest to be restored? and how we, using a different methodology approach to accelerate this natural succession, and eventually we back to the original rainforest. So based on the literature, they have two broadly perspective. One is small area, low intensity, frequency logging and deforestation. I think it's possible and restorable because the soil environment, the seed salt for species regeneration is there. But if you have the large area and high intensity frequency disturbance, and it could be limitedly restorable because the soil, the topsoil losses, appropriate engineering approach and the pioneer species have to apply. But we could wait for so long time, eventually back to original one, but even we are not very pretty sure. But another one could be unrestorable in the certain time. Even in our lifetime, we can't see really the natural rainforest that come back to original state. So this is the point. And I give the one example and published in the Science 2021, and the example showing in Brazil Amazon rainforest, once the original local perimeter forest log and then replaced by the eucalyptus species, and that leading the 86 species diversity loss. And even though after 14 and 90 years later, the number of species still less than original forest, particularly they counted the bird species, and bird species composition changes, and they give our indication it's very difficult to back to original state. And another paper also saying that lightly distributed tropical forest is relatively easy, but it takes 10 years to recover soil, 20 years to resume the plant function, and 20 to 60 years to rebuild communities structure, and 120 years maybe, yeah. But for us, we cannot see really we have reached that level on the restored function, particularly the biomass species composition. Now I talk about in China, through over the nearly 20 years of our practical experience, and then try to linking with the ecological theory, what we understand, we put up so-called rare species regulation mechanism. That means we identify the rare species, one of the key species group on shaping the ecosystem function and then ecological restoration. And the ratio of rare species in tropical forests in the range of, sorry, that is back in this, the shade area is 20 to 50. And we think the rare species serve the key species or indicator species. That's very important, shaping the recovery of species abundance and later on the recovery quality. Quality means structure and function. And rare species, and also we call the common species, constitute the mosaic pattern of the coexistence of diversified habitat. And the two kind of group of species actually use the different habitat. The rare species try to have the clamped distribution along the soil gradient with the increasing phosphorus content. And then the common species really associated with the increasing nitrogen availability. And if we using this sort of a framework, we need to think about one is interspecific non-random competition, and then the microclimate appreciation of preference, and then the plant soil feedback based microbial mediation. all these three dimensions, and then we try to put together to come up this idea and then apply in the real world. This is an example showing the tropical forest restoration with different logging. And after 50 years, you will see the Heliophilus species were reduced, and commonly we can see that. But shade-tolerant species, we call particularly the rare species, were gradually recovered, but we're still lacking of the key stone species in the late successional tropical forest. So even though 70 to 80 of species richness restored, but in terms of the species composition, structure, and function, it is far from being restored and compared with the original primary old growth. And they're also showing, along the temperature scale, we actually measure the 3 time point. One is 410, and then 15, and then 2020. You will see the early stage of the secondary and the middle age of secondary, and then old growth. You will see, based on this long-term monitoring data, and the lack of the original forest species, lead this succession stage is very slow. So, and also we counted the distance of the similarity of the species composition compared with the original old growth forest. And they give us the same story. So the species composition is very important. So under this situation, we think we have to put up the active restoration. Active restoration means in forestry practice, we're using the thinning and then select cutting, and plus with the replanting of some species. Here, the species I call the rare species, and particularly for those species in the later stage of the ecological restoration of the rainforest. So, for example, the Hopia financis is a feeder species of this tropical mountain rainforest. So once you, through the thinning and encourage the natural regeneration of such the species, and also replanting, once the natural regeneration is could not be possible, so +2, and then you really can accelerate this process. So dealing with the different management option, one is selective logging, and also clear cut. You will see, for the selected logging is relatively easy, particularly for the rare species recovery back up. For the clear cut, it take a longer time. So based on those measurements and then historical recorder, we can understanding the rare and the endemic species in the mountain rainforest really take much more time for restoration and after the logging. Yeah. And so based on this idea, we try to understand which the rare species and playing the key role in the whole restoration process. We do lots of field work, and then some species in the early succession state, you have to remove that. And then later on through the thinning and then replanting those of the rare species, and then we accelerate those recovery rates of the restoration quality of the community. Anyway, so later on, we think once the rare species are better restored and then increasing their population size, and eventually it's better. But for the whole forest landscape, you have to identify where we put up we call the plant functional group, and using this sort of idea to try to simplify the complexity and the structure of the completed highly diversified mountain rainforest, and using those units, and then we can applicable to the real field and restore those and along the environmental gradient, and also different community characteristic. Now I come to my final conclusion for the secondary forest formed by the large-scale clear cut resulting from the conventional slash and burning practice. We really need to active restoration in order to accelerate the recovery reach and to reach the optimal species composition stand structure of intact old growth forest. If you don't do the active restoration, I think it's really take long time and then back to original. So that is necessary. Assisted active restoration method like the thinning, thinning, and replanting demonstrate an effective approach to improve regeneration and the rejuvenation of the key ecological function species, and will achieve the synergetic enhancement of biodiversity and ecosystem function. And rare species play an important role in shaping species richness, recovery, velocity, and quality, and during the ecological restoration of degraded tropical rainforest. And finally, if we applying the substitution of plant, the functional group is the effective way and for the tropical forest landscape restoration, and then they help improving restoration efficiency and performance at the multiple scale, as I said, species level and community level and landscape level. We need to figure out exactly efficient way and to take after our degraded forest and back to original forest state. Oh, thank you. I hope I finish my talk within the time frame. Okay, thank you.
Thank you very much, Dr. Liu. Yes, you were very prompted. Those were beautiful pictures. Oh, we have another one. Yeah, would anyone like to ask a question? Because we have a few minutes. Anyone? I'm going to ask a question, but I don't want to interrupt you. So in the, oh, sorry, you have a, yes.
Okay.
Thank you very much, Professor Liu, for this very nice speech. I enjoyed it very much. So I have a question in terms of restoration. You say the assisted restoration measures could be a better way compared to nature restoration. What do you think about that?
Yes, I fully agree with your opinion and concept. We have to follow the natural successional pathway. And then the here idea is that we really examine the whole process of natural regeneration and succession, how this is going after degraded, the point that here I try to emphasize how we accelerate those natural succession, within our lifetime, and eventually we can see that's really back to original state of the primary rainforest. This is the point. So we wouldn't have too much of an intervention with the human and change the whole structure and the process of ecosystem. And we try to stimulate, just stimulate this process.
Okay, thank you.
Okay.
Yes, Ben Turner.
The rare species on Hainan, you mentioned that there were a couple of Podocarpus or Podocarpaceae on there, Dacrydium and is it a Dacrycarpus? they tend to be relatively rare in tropical forests anyway. So I wondered how you go about encouraging those species back into reforested landscapes. It must be quite difficult.
Yeah.
Is it a soil thing or a active planting or?
Yeah, it is. The first you need to have a large area of field investigation and to see, where and rare species located, and you will examine the site condition. For example, some soil chemical and physical characteristic, and also the surrounding the trees, how they make their cluster or groups. And then once you have this information, and you will using this sort of the knowledge and then put into the real field. But in the real field, just as I said, the large area clear out. Sometimes you cannot actually have the seesaw of this rare species. And we use the wild plant in the summer area and collect their seed and put into the seesaw and then cultivate it, and eventually like the small seedling, and then we reintroduce in the field. But anyway, once they have the natural the situation and still have the seesaw, and we encourage that using the very small thinning and select cutting and remove some of the species surrounding area. As I said, a pioneer species, they really have the function can constrain their regeneration at the early stage. So this is a problem. So we do some of that. But this is the demonstration pilot project. You cannot do the whole area everywhere, yeah. But depending on the human, capacity, anyway, we do that step by step.
So in the active restoration, I'm interested in rare species. I've studied rare species a lot. And it occurs to me that I could state that species abundances are utterly random. And what we're seeing right now is a snapshot of a community that's been drifting, and the species are rare. So if I were to encourage rare species by planting more, I would actually be changing the community, because those rare species would become abundant, while abundant species would become rare.
Yes, you're right. We cannot do that over, natural distribution pattern. You will know exactly, how many, the number of species and their spatial heterogeneity's distribution, and we need to know sort of the knowledge. But for me, some of the rare species, as I understand it, is very conservative. It's not very easy, once you replant it and encourage it to generate that regeneration, you cannot quickly spread, Is another biological limitation for this species, why he have this random. So we wouldn't want to change the whole structure. It just simulate the original community. Yeah.
Well, this would be a big experiment I would do. Is I'd clear Barrow, Colorado Island and Panama, come back in 300 years and see whether the abundances are all exactly the same or whether they've all changed. I'm not sure what would happen.
Well, for me, we cannot predict it for a long time anyway. This is the way, we can do that right now.
Thank you. Those were beautiful presentation and beautiful maps of Hainan. Thank you very much.
Well, thank you. And we hope we have a chance to visit the real site. Okay, later on.
We will.
Okay, thank you.
Dr. Kitajima, you have her presentation ready. So I'm honored and very pleased that Kaoru Kitajima agreed to come here. I've worked with her for many, many years. She's been a expert on tree species at Barrow, Colorado Island and Panama. She's learned many valuable insights about them for over 30 years. She originally, she's from Japan, studied in Japan, then went to Illinois, of all places, where I live now, to get a graduate degree before she took up a professorship in Florida. And that's when she started, well, in Illinois, that's where you started work in Panama because of Carol Augsburger. But Kaoru is going to take on we're going to go on a very long journey to a very different forest. And one of the most different forests I think there is in the world, in the tropics. And she's going to tell you about this place in Madagascar. Thank you, Kaoru.
One is that. Okay, right. Thank you very much for a very kind introduction and invitation. I have known Rick for a long time, and I'd like to thank the Beijing Forestry, University of Beijing Forestry, and also the editors of the Journal Forest Ecosystems. So I will take you to Madagascar, and one thing I want you to pay attention is this is the forest I will take you to and explain more. Where is Madagascar? It's in the east coast of Africa, across from Tanzania, across Mozambique Channel, and the site is in this red dot in northwestern Madagascar. And it is a white sand forest, as you see this image, very gentle slope, looks almost flat. And a star to keep this forest going is right in the middle, brown lemur. Now, just before that, as Rick kindly introduced, I don't have to tell much. I am professor of tropical forest ecology and environment in the Graduate School of Agriculture, Kyoto University. As many of us seniors do, I also work in the International Strategy Office of Kyoto University at large. Also, I am currently the president of the Ecological Society of Japan for one more year. So I want to do a little bit of advertisement. So Ecological Society of Japan has a long history. I really would like you to look for it. And we hold annual meetings in March with sessions in both in Japanese and English. And this year, in July 2025, okay, right, thank you for smiling. We will host the biannual meeting of the East Asian Federation of Ecological Societies of China, Korea, and Japan. So yes, you can look for efest2025.com, and you can still register to attend poster sessions, but participants are limited to a total of 400. So please rush. But I just want to mention that of all the 20 symposia, can I use this? There are some the pointer center. Okay, back. So there are symposia including several that address forest ecosystems, including response of forest ecosystem to geographical and temporal environmental changes in East Asia. Symposium L, the mechanism underlying variation in forest structure and the maintenance of ecosystem functions, and symposium, and another symposium, sustainability and carbon sink of plantation forest ecosystems. So all these symposia are already planned, but the symposium organizers may be willing to accommodate a few people who might be interested in a particular symposium. So please visit our site. And so now I want to bring back to tropical forest. Of course, I'm familiar with the Forest Global Ecosystem Observatory run by formerly called CTFS, as many of you are familiar. And there are 29 countries that have total 29 sites, but no large-scale forest plot in Madagascar. And this another thing is very few tropical dry forests. One of the oldest, fourth oldest large-scale, monitoring plot is Mudmalai of India. Another one is one of the newest addition to the network, San Emilio. So what I will tell you is our ambition to be one of the 3 dry forest sites in Forest Geo Network. We are working on it. I'll get to that. But first of all, what is Madagascar? Biologically, it is very unique with lots of endemic species. It's the 4th largest island in the world with independent biological evolution after separation from Africa 100 million years ago during Cretaceous, when the Gondwana split into major continents. So Madagascar is a chunk of Africa, but just as ancient as Australia in terms of unique biological history. And so Madagascar forests have really high level endemism for animals, plants 96% at the species level, 96% for amphibian, 91% reptiles, 51% of birds because they can fly from Africa, but mammals 93%. And so there are no monkeys, instead there are lemurs of many species occupying different niches of different sizes, from tiny mouse lemur that can be on your hand to relatively large-bodied cat-sides, and some extinct really large lemurs. Because of the trade wind, rainforests are all in the east coast that brings in rain over the ocean, Indian Ocean. Ankara-Fantsika National Park is the right here. I think we are running out of the juice for pointer. But dry forest is in the northwest, and it gets really dry. There's no more forest in the southwest. So the focus for my talk is, oops, I can do this, this northwestern dry forest. But I want to talk about geology. So this, I told you, it's a white sand forest. And where did the sand come from? It's ancient sand dune from Jurassic time. Indeed, there are dinosaurs fossils coming from this area, very famous as Majungasaurus. Majunga is a little bay at the left, upper left. And what I want you to pay attention is major difference in the geological substrate. Eastern side is like ancient continental shield, but west side has the 100 to 200 million years old sedimentary area. So our site, Ankara-Fantsika National Park, located about here, is at the top of so-called Ankara-Fantsika Massif. That basically the sediment accumulated in very shallow ocean and became land even before split of Madagascar. So this is a kind of sedimental substrate. There are some lateritic, very hard surfaced one, which protect loose sediment underneath. And these are hills that are raised. And places you can see some white sand. Where this white sand is exposed, it becomes a gentle hills like at our site. So northwestern dry forests have had the highest loss rate of forest in Madagascar. So I think a lot of times dry forests are less focused than rainforests in terms of total biodiversity. Number of species, yes. And the carbon stock, right. It's less than in rainforests, but it has unique biological diversity that needs to be conserved. But northwestern Madagascar has higher loss rate of original forest. By the way, humans arrived in Madagascar less than 1,000 years ago. So that really makes difference. In the rest of the world, we don't have dry forest without modification by humans. But here we have pristine dry forest that was very little damaged or modified by people. And so, but it's been threatened. It holds one of the largest remaining dry forests in northwest Madagascar as one of the first 5 protected areas established in 1927. But you can see the hotspots around Ankara-Fantsika is very dense. Every year there is a fire threat coming. But there are still very nice forest, original forest here. Angra Francisca National Park has a very strong seasonality with dry season, with almost no rain for 6 months or almost 7 months. My abstract is wrong, not over 7 months, almost 7 months. Yet this seasonally dry forest is only partially deciduous. So the picture at the left, which is rainy season, at the same spot you see this tree incline, and during the dry season, you can see a lot of evergreen trees. And this is the kind of strategy the previous speaker mentioned, more tolerant strategy of infertile white sand. Now, so Angra Francisca National Park has really 135,000 hectares. It has really nice land planning starting in 1927, and they have designated research area in which you can do biodiversity survey. Crisscross of trails allow people working on birds and mammals to walk around and monitor. And we decided to establish a 15-hectare plot here in the middle of this designated area for scientific research. So this is a picture of the team at the beginning in 2019. And you see, some of you may be familiar, Dr. Yifan Sun of Donghua University. I believe he was advisor to some of the large-scale plots here in China too. But he has plots in Taiwan. So he came to give us advice. But one important person, Hiroki Sato, here in the center, he's my closest colleague and next generation. But he's worked here at this research site for over 20 years. He started as a Lima researcher. And he approached me, hey, can we collaborate? So I said, hey, let's start a forest geostyle plot. So that's how it started in 2019. And some of our earliest result is found in Fujimoto et al. 2024 ecological research published by Ecological Society of Japan. So we did the tagging, marking, measuring, and species ID, and mapped the location of all stems and marked. This is 4 or 5 greater, diameter at breast height greater than 5 centimeters for the entire 15 hectare. And we did make a lot of, voucher specimen, made a picture guide that could be shared with the local stakeholders and students. And after 2022, in 2022, we started to map for the center of 6 hectares, 200 by 300 meters to map everything for greater than 1 centimeter in diameter. So this resulted in terms of 5, we have a better data cleaning for species, for stems greater than 5 centimeter in diameter, 147 species, mean DBH 8.8 centimeter, maximum is 69 centimeter diameter, but mean height is only 8.9 meter, and maximum height is 15.6 meter. This is pristine dry forest, but on white sand soil. And 29% of biomass is made up of trees dispersed by Lima. That's something I'll come back to. In terms of the data from the central 6 hectare, if we start to talk total, including small stems greater than 1 centimeter diameter, we have 12,517 stems per hectare. This density is much higher than San Emilio, Costa Rica, which is largely secondary, and Mundmala, India, also very severely disturbed secondary forest type place. So we have extremely dense forest, but the 15 hectare is large enough to do this monitoring in terms of the species area curve here. We have mapped the trees and lianas. So liana is a minor component, but they are important as in many other tropical dry forests. And you can see many of the stems are very small, but a few can be larger, both in terms of stem number and species. These are the common, most abundant, dominant 20 species, which occupy 78% of the total base area. And I cannot agree more about the importance of 127 other species as well. However, important point is all the dominant species are, I mean, not all, many or most of the dominant species of evergreen. So leaf phenology in terms of evergreen or deciduous, here we distinguish not just the straightforward evergreen, but also late deciduous, which keep the leaves until late in the dry season. So it's a brief, brave deciduous kind of thing. Progressively deciduous keep on losing leaves. Perhaps the drought stress of the dry season is heavy, so they lose some leaves, but not all. And we still have the ones we haven't been able to figure out the phenology out of all the 147 species. But this is, weighted for the number of individual and basal area. But the clear pattern is evergreen species dominant. So if you look at the distribution of species here, I'm also, these maps of the top 16 most abundant species, there is the contour of the topography. Lowest point of our plot is 150 meter above sea level, up to 195 meter above sea level. And some species you see clear distinction. This one, the most abundant one, and second most abundant one, you can see this species likes the lower slope, this species likes higher slope. Not just that. If we plot the community-wide thing, and so this is each point is a data for 10 by 10, 10 meter by 10 meter by plot. And then we can plot how basal area diameter, maximum diameter, median diameter, which are the indicators of forest structure, change with elevation from low to high. So you can see the trees get smaller in perhaps more stressful higher elevation, which tends to get dry. But what's really funny is number of individuals, number of species, and species per individual actually increase with elevation. So rare species start to do better in more stressful environment. In terms of Shannon's index, similar. Simpson's index didn't work, but Fisher's alpha too. So maybe I forgot to say. So higher density of smaller and shorter stems, but higher species diversity at higher elevation. But when we look at the eyes, we can't see difference. We could detect it only after having a plot and actually enumerating. And once we have this kind of community-wide tree species data, we can evaluate quantitatively how important the seed disperser animals are. So this is a brown lemur, the largest lemur species at our site. And they disperse, they're the exclusive disperser of large fruited, large seeded species like Abramea deflexa. So Hiroki Sato actually analyzed the feces of lemur and identified the species. And from that, he could tell which species of trees in this tree community is dispersed by brown lemur in terms of number of percentage of total individual, all the individuals, percentage of basal area, and number of species. So the dominant species are dispersed by brown lemur. And especially these are the species that can be dispersed only by brown lemur because they are large-seeded species. Now, so here is a star, brown lemur, and you see my fingertip? It's very coarse, quartz-rich white sand. This is where local people sneak in and dig up yam, Dioscorea tuber, and you can see all the sand coming out. It's pure white. And so big fruit with big seed dispersed by brown lemur, they do pretty well during the rainy season. A lot of them germinate. Towards the end of the rainy season, you see very good seedling regeneration here. What keeps this ecosystem with loose white sand going is the secret is in the root. It maintains species-rich forest on the top of white sand hills. And this is Tomohiro Nishigaki of JIRCAS, Japan International Research for Agricultural Science. And So we dug soil, and you can see a lot of root mat close to the top, but loose white sands at the bottom. But when you magnify these roots, you can see a lot of sand grains are clinging to fine roots, and moisture remains even at the end of 6-month-long dry season at the root mat layer. So root mat keeps and recycling moisture to keep this forest going to support evergreenness. The trees do not have deep roots, but once in a while when you dig, you find some roots going farther down. And these are liana roots, in my opinion. We need to verify, but you can tell, cut the root, and you can tell it's liana or not. So dense root mat you can even pick up. So we analyze the root. But when we started digging roots, about that time, about 2022, this is during COVID-19 pandemic. We couldn't go to the site. Our assistants called us by phone and, hey, fire's coming to our plot. So here's our 15-hectare plot, and here's a fire that came so close, only 30, 40 meters from the corner of our plot in 2021. This is the area burned for the first time. You can see the trees, many dead trees, and the exposure of white sand. Extremely vulnerable. So this is published in PLOS ONE by Percival et al. And he also went back to the fire history using Landsat images. And you can see there is no evidence of fire in the area surrounding our plot, 3,000 hectares, up until 2014, just a little bit, 2014. Oh, and the same area burns again. So previously burnt site, forests tend to be burnt again. So it starts to accelerate the burning cycle to destroy. So this is a map of the area burnt in 2017 for the first time. And yellow, blue is the area burnt in 2019. You can see a lot of overlap, and the same area burnt again. So like every 2 years, why? You can see if you look at the picture, the first burn keeps a lot of tree stems still standing. Some of them are alive, try to resprout, some of them are dead. But the left side burnt, right side unburnt, thanks to the trail that worked as, fire break. But these dead trees becomes a fuel to fuel next fire. So once it's burnt, it is prone to burn again, killing even the surviving big trees. And after 2 fires, it turns into desert. How can we restore a place like this? It's a lot easier just to keep the forest protected from fire, but we are also thinking about restoration technique and how the post-burn vegetation might recover after first burn and second burn. This is done by Andu Rakotomamonji, who is a Malakasi student now, analyzing her data for PhD. Her data consists of 21 plots of 5 meter by 5 meter in natural forest and area burnt once, area burnt twice. We kind of can make this transect using the, well, we didn't want fire, but since it happened, let's make the best out of it. Now, so a lot of trees are resprouting after first burn, after second burn. As I said, it turns in desert in this kind of thing. So we monitor what is the ground condition and soil and roots, etc. Come on. So the soil profile photos show disappearance of root mat. Natural forest, root mat down to 20 centimeter, becomes very shallow and sporadic, and after 2 or 3 burns, almost no roots. When there is no roots, there is no recovery of vegetation, because this is not a tropical dry forest already modified by humans for 50, 60,000 years that may contain more species that are fire tolerant. So these species are not fire tolerant mostly. So here is a root biomass, living roots in green, necromass, dead roots in red, and root phosphorus pools again here. So the root mat in the top 20 centimeter disappear mostly after first burn, but there is a little bit remaining. Second burn kills it all. Coarse root in 20 to 100 centimeter, it's deeper. Some roots are going down. There is some live roots, lianas included, and these can accelerate the recovery, but second burn can kill it. So these live roots after first burn disappear mostly. And phosphorus in root, this is the phosphorus stock in gram of phosphorus per square meter of ground down to 20 centimeter. A lot contained in live roots disappear as well. So we really need to have on-the-ground conservation. And here are two of our local assistants, and Ruba on the right, he's a doctoral student at the University of Majanga working with us. And we decided, let's make a fire break. So 6-meter fire break was created in the middle of the area that we experienced one burn. So we keep on maintaining every year, got to protect it. But just to switch the why we have to protect this dry forest. It has lower carbon stocks, it has lower diversity than rainforest, but many tree species in this forest provide diverse ecosystem services to local people. So Sato also did it in a botanical interview with local people using the picture guidebook we took. Do you know this species? Do you know this species? What do you use it for? For medicine or carpentry or rituals. And you can find, okay, sorry, it's building material tools, ritual, cultural, medicine, etc. When you combine 80 to 90% of the stems or the species are used by local people. Very, very high for any forest. And forest loss by fire impacts ecosystem services at watershed level, much larger scale. Because you can see here is similar to the picture in Hainan, but it happened a little more recently. 2010 to 2021 development of agricultural land, and this resulting exposure of soil and sedimentation coming to bury the rice field in the river, along the river. And when there is more sand piling up on top of the original, more organic matter-rich soil, you lose the rice yield. Sorry, this is not in Chinese, but it says rice yield. And this is the depth of the sand accumulated. So this is the soil erosion monitoring plot. So this is the amount of soil eroded from burnt forest on slope and similar slope natural forest, almost no erosion. So forests provide critical ecosystem service in preventing soil erosion and sedimentation, and protect the water storage in hills, and protect rice production. So there is a landscape level forest conservation to be combined with food systems sustainability. So this is, we have a sandy soil, sandy hill, which has a high infiltration capacity, but the tree roots and the vegetation protect the surface runoff. So a lot of infiltration of water, and this whole Anchor-Fancy massive hills becomes water tank to provide water downstream into a large rice field region, which has irrigation canal. Second largest rice production region in Madagascar. When the forest cover is lost, it also means loss of infiltration capacity, and we will be monitoring this. This is a new applied research we have started. But watershed level perspective is very important too. So here is actual drone view of our site, the area burnt in 2021, and intact forest is Lake Lavalova, famous for bird watchers. So where do we lose forest first? because we start to cut forest and turn in agricultural land on good soil first. So some of the remaining natural forests are found in infertile, nasty soils. But these are important for keeping natural biodiversity. Such forests may not hold much carbon, but they conserve high biological diversity and ecosystem services, including regulation of water supply and at local and regional levels. Ultimately, such natural forests should be conserved for a sustainable landscape that balance food production and biodiversity conservation. And some of the data I presented are found in the recent publications and also yet to be published master's thesis. But we have many more data to come, and we are busily summarizing the data for all the stems greater than 1 centimeter so that we can join CTFS network. Thank you very much.
Thank you very much. That's a really spectacular study of a forest, and a remarkable forest that is being threatened by that fire. Someone want to ask a question? Otherwise, I'm going to take up all our time. Dr. Ma.
Thank you very much. Excellent presentation. I really have interest to know more about what happened in Madagascar because I haven't been there so far. And I just asked a question related to this picture. And you can see the burned area and the unburned area, which is, say, the burned area is very close to your 15 hectares plot. And my question is, there's no natural barrier between the burned area and natural forest. And what is the reason the fire just stopped in such a boundary? Thank you.
Thank you very much for excellent question. The fire stopped because it may not be visible, but there are trail systems that people can walk in, and the park officials and our assistants too, when the fire started to encroach our site, they rushed to the site, use the trail system to put the fire. And that's why, fire break is very important, but at the same time, the population surrounding the Angkor National Park is increasing. So when the forest that was buffering outside of the park boundary was lost, that's when boundary was impossible to keep, because there is no road at the park boundary. So local people, some of them know, some of them don't. And actually, when the park was established in 1927, the policy was to allow the local people who are already inside the park to stay there, but make a contract so that they cannot expand the rice field, and they have to honor the park's rule about community forest usage and a strictly protected area. Communities inside the national park understand that. However, increasing population outside the park is putting pressure. They are even jealous, envious of stakeholders inside the park who have access to community forest. So they are outsiders encroaching and doing, sometimes set fire, doing the, bringing cattle in. It's just very difficult political situation. So we want to provide strong evidence for that. It's really important for food security too. That way, hopefully, Malagasy government will be able to give some more policy. Immigrants are an important issue. Climate change in southwestern part of Madagascar is really driving people to migrate within Madagascar. And that kind of, pressure is very, very difficult to cope with. So thank you. It's a long answer to your question, but then they don't necessarily honor the boundary of the park, and there is no very clear, fire break. But fire break at the more refined scales, strategically placed, could be useful. Right now, each community and village has fire breaks surrounding the area, but it's not working very well. Thank you.
Thank you.
Yes, another question.
Okay, I have two questions. The first one is you mentioned that the forest carbon there may be not the top priority, but I think the carbon density there seems really high. How much was the carbon density in this forest? Is the first question.
Higher carbon stocks. So, but we cannot just argue with carbon stock. How do we actually factor in biodiversity as a reason for, protecting forest is something many of us have to think about.
Okay. The second quick question is that we see that the forest that is being fired and there's white sand on the top. So have you been touched to the way to restore this forest? Because it could be quite different because the sand seems not easy to restore these forests.
Right. So coming back to, I think maybe this is good. So this is our 15-hectare plot. This is the area burnt. And this is Andrew's fire impact monitoring plot. She's comparing once burnt, twice burnt, and never burnt. Okay. And along here, this student, he is growing native species in the forest and outside the forest, and he's doing transplanting to see if we can do the accelerated restoration. So we are working on that too. So my hunch, we have to see, is that the once burnt area would be restored by accelerated planting of white sand forest species. But the twice burnt area may be already too stressful for even that kind of species. In that case, we need to combination of pioneer species that quickly cover, provide ground cover, and into which we can transplant some of the natural forest species. We are experimenting, but it's his doctoral thesis work that he's working currently busy in the field.
Okay, thanks.
Caruso, I'm going to ask a question first by assuming that the fire was actually ignited by people.
Right.
And is there lightning in the area? That was, lightning fires don't
No
happen.
lightning. So the analysis by Josefa Percival, oops, I think I will go. He also, as part of his doctoral thesis, he also analyzed the event that caused forest degradation and forest recovery. And so there are cyclones, but to cyclone impact, it's in this much larger area. So mostly human impact, and the fires are more frequent, closer to the people, human communities, etc. But there is lightning is extremely rare here. They are cyclones, but never heard of lightning strike.
Okay. Yes, ask a question.
So thank you very much for your great presentation. I just wonder, you showed the soil over there is pretty sandy, but you also showed us some soil profiles, but they look not that sandy. So I just wonder, the spatial variability of the soil properties over there.
Yeah.
Is this typical, or the sandy one is typical? Thank you.
So one thing, I think this is a scale that matters for trees and lianas, and where the forest exists, there is a root mat, and below which is white sand. However, I showed you earlier the geological substrate of the site, this, right? So much larger scale, where you find forest is where white sand is on top of this red hard pan. Who knows? We are talking about the time of dinosaurs. And then this hard pan, reddish, sandy soil, but it has a very hard crust at the top, lateritic crust. And it's so hard, I can pound on his hammer and it wouldn't break. So it can keep the water from breaking, but it's like eggshell. This is a vantage point. You can see that here suddenly, if the top shell collapses, huge erosion. They call it lavaca here. It's the big erosion, scale. And you can see the profile of, we are talking about Jurassic sediments here, all piled up in shallow ocean. But depending on the river and the type of sediment that comes in, there is different textures, but largely loose, unconsolidated rock, sedimentary substrate. So it depends. So this is natural vegetation, in my opinion. It's not degraded savanna. It's a natural savanna because the soil is shallow and its exposure very flat, lateritic hard pan. I think there's no point of trying to put forest in here. This is natural grassland savanna. But this is natural forest on white sand. This is the forest to protect. So we really need to pay attention what the original vegetation was. That way we can bring out the best of the power of nature, nature-based ecological sustainability. I think we need to pay attention what was the original vegetation. But thank you for the question.
Thank you very much, Kaoru. So we're going to have 20 minutes for your discussions to continue, and I think we have coffee outside, is that right? And we'll be back at 10:40 for the next lecture, 20 minutes from now. Thank you very much.
Thank you very much for your attention.
谢谢。Yeah, I will go.
Pilsun's going to speak.
Yeah, after you, I will just use it. Okay. 好。
Okay, then. I have the pleasure of introducing one of the members of the board of the journal, Forest Ecosystems, Dr. Pilsun Park. She's from Seoul University in South Korea. She studied, I didn't know this when I communicated with her. She did her PhD in the United States after studying in Seoul as an undergraduate and master's. She went to the United States and worked at the University of Washington exactly when I was there. So we probably met or passed each other in 1999, but I didn't know she was a board member of Forest Ecosystems back then. I wasn't a board member. Well, there was no journal, of course. Two future board members of Forest Ecosystems were passing in the hallway, and what's the name of that building? Winkenwerder. Dr. Park came back to Korea after finishing her PhD at University of Washington, worked with the government for several years, and then took a job at Seoul National University. And so I'm pleased to invite her to speak about forest restoration in the tropics.
Thank you, Richard. Okay. Hello, my name is Pilsun Park, and then I would like to take this chance to deliver my thanks to host this wonderful workshop and then treat me well, like Osbert, John, and Richard, and all my colleagues. And also, I really thank Beijing Forestry University to host me last year and this year. I really enjoyed the workshop last year. So when Richard emailed me to come, and then I said yes. And then this is the midterm period in my university. So I just let my student take exam, and then I'm here. So I'm from Korea, famous for forest restoration. So you probably have heard a lot about forest recovery and forest restoration in Korea. And today I'm gonna talk about like some part in Korea, but also the lessons I learned when I visited tropical forests. And just before this, I start my talk, I also would like to announce the workshop conference that I'm preparing in September in Jeju Island. So if you visit this website, cccf2025.com, and then we are hosting a conference on conifers, because there are a lot of conifers, especially at high elevation, conifers are dying, probably due to climate change and also anthropogenic activities. So the registration is still open. So if anybody who is working on conifer forest or conifers, you're welcome to participate. And the place is especially in Jeju Island. So for Chinese colleagues, you don't need visa to come to Jeju Island. So please consider to visit us in September. Thank you. And then I will go back to my talk. Okay, so we all know, and I'm so glad I am talking next to Professor Liu Shirong, and then also the professor from Japan, because they covered a lot of topics that I was planning to talk. So they all pointed out all the importance in tropical forests and then the restoration issues. So we all know that there are a lot of areas, especially the global forests, are destroying and being destroyed. and we lost about 420 million hectares of forest for the last 30 years. And it's United Nations statistics that the world lost more than 10 million hectares per year for last 30 years. And I believe that it's only because we have statistics for the last 30 years. So we might lost more than that, but we did not have enough data on it. So and in Korea, South Korean forests are about 6.4 million hectares total. So which means the world is losing forests 1.5 times larger than the whole South Korean forest, and it's still ongoing. So, but when we compare them with the 1990 to 2000 and 2000 to 2010, so the rate of forest loss is decreasing, but still it's ongoing. And then when the United Nations published all the data about it, and then I just copy them. So it's natural, but the problem is that we are increasing the plantations. So we are planting trees. But the thing is that we are still losing natural forests. So, this decade is UN decades of restoration. So there are all like restoration is big issue in all over the world. And the restoration, there is marine restoration going on and forest restoration going on. And if you visit the website of United Nations, and then it says the forest occupies more than half of the restoration spot at this moment. So it's all like, we all know the importance of forest restoration, and we all know that we need to increase the forests. But so forest restoration is a key issue in all environmental discussions, and most of people know that. But the thing is that we all know that restoration provides all like good ecosystem services, but we still lack of data on how exactly the concrete data on how much it was improved after restoration in those things. So we are doing studying. And when they compare the restoration and forest increase and number of forest areas, and Asia is doing quite well, because when we compare the data for 30 years, and then like Africa and then South America lost a lot of forest areas, while only Europe and then Asia has changed from lost to increased in forest area. And especially when we look at the Asia, it increased quite large areas. So we could think that, oh, Asia is doing well, and then we are doing good job in increasing forest area and forest recovery. But when we look at this data, this is for each decade from 1990 to 2000, 2000 to 2010. So we all know that if you see the upper part is increase and the lower part is decrease in forest areas. And like Asia is doing better job than Europe, but it's because Europe, most areas are developed, and then the rest of them are, quite protected than other regions. But South America and Africa are still losing quite large areas. And this is the map of changes in forest areas, and red means we are losing forest areas, while green parts are the places where forests are increasing. And Asia was doing well, so we are increasing the forest area, but that was only thanks to China and Vietnam. So except China and Vietnam, most of Asian countries are still losing forest areas. So, and especially the tropical countries are losing large area of forests. So a lot of people going to Southeast Asian countries and Africa, and a lot of money, especially like United Nations put, and then also all the ODA funds are going to the tropical region for forest restoration. And for example, I will talk about it later. So we are doing a lot of work and efforts for forest restoration and increased forest. And then I've been doing for this work for probably for last 20 years. That's quite less than my colleagues sitting here. So, but I probably would be mid-career stage at this point. And then today I would like to talk about like 3 lessons I learned from the areas that I visited. So sometimes we are confused with restoration and rehabilitation. And we used to use rehabilitation, but now we are using restoration. But lots of forests are going to restoration once it was rehabilitated. So these days, restoration kind of used more in the widely concept. So a lot of people just use restoration. But there are a lot of areas were disturbed by anthropogenic or other natural causes. And then there is some succession going on. So like Dr. Kaori once showed us in Madagascar case. So there are areas that are just going naturally recovered. But this is a very typical restoration, the direction diagram, but I got a little bit changed it. So, but if it is just let it there, and then it could be just go in some direction. But we are not sure it was its original condition or not. So there are people actively engaged, or there are some issues, still a lot of big issues going on with just to let it be there, or do we need any active actions to make this forest restored? And when first forests were destroyed, and then when we try to do something, either it's let it be there or just to do actions, we could see that there are physical conditions are changed. So when forest was tried to get recovered, first it was a physical condition, and then later it's vegetation coming in. But that vegetation was not the vegetation from the original state. Usually like old growth has to come later. So the pioneer species coming in and they change the physical conditions. And then later when forest gets more mature, the ecosystem services can be functioned more fully. And then what I witnessed is that first change could found out in the more like physical condition, like especially in the temperature or humidity at the air or in the soil. So I will just point out each items. So this is the place. The first, I learned 3 lessons. That first, the climate regulation is really big benefit of forest restoration. So this is the photo of it's in Korea, and it was mining field. So they stopped mining, and then they just try to recover the area by planting trees. And then when we compare the 3 areas, one is like barren. So it was just finished mining. So nothing happened. They just put the stones there. And then this start is somehow the mining stopped about 5 years ago. So the seeds coming in, and then they tried to establish there. So we collected the soil temperature between the bare land and then the bare land, but under the tree, evaded the area. And then this is the area where rehabilitation was conducted in 2009. And then this green line is the soil temperature in natural forest adjacent the field. So the whole area was within like 5 kilometers in the boundary. So they have same climate. And then their soil was very similar before. And then the third thing we shocked was that in the bare land where the stones were accumulated, and then in the summer, the temperature went all the way to the 60 Celsius degree because it was the soil temperature. There was actually just stones. So we could not get the actual soil temperature. So we just pushed our sensor into the below 5 centimeter and then just let it there. But because the stones were, it's a mining field, so stones were black. So maybe they kind of observe the light. So it goes all the way to the 60 degree. And then that means that trees, especially seedlings, it's too hot for them to survive. So while this natural forest or the area where about 15 years ago it was rehabilitated, soil temperature was quite well stabilized. So it was only like 15 years different rehabilitation, but still once the tree covers, and then we could witness that the soil temperature became improved much better. So this is the air temperature. Left side is air temperature, and right side is our soil temperature. And then when we also see, actually the air temperature was not much different between the natural forest or the bare land or rehabilitation sites, because the air just move around through wind. But soil temperature showed quite differences. That is, the above side in the right side is the average soil temperature, and the below is the maximum soil temperature. So this is the same photo I just showed. So you can see in the summer, we just measure for 2 years. And then in the summer is the temperature difference much higher. So and then if we just let it be there, and then there's very little chance for seedlings. There are a lot of seeds coming in because the outages in the forest send a lot of seeds disperser. But still, because the environment, especially soil conditions, is so difficult for them to survive. So they just germinate, but they all died. And then this is the relative air humidity in the left side and in the same site. But the left side is left photo shows the relative air humidity, and also the right side is soil water content. And we use the relative soil content, and then the upper is the bare land, and the middle is in the rehabilitation site, and then the lowest is the natural forest. And when you see the annual mean relative humidity is in the natural forest is about 9%, while there is we did not show the bare site, but still the rehabilitation site is 70%. But in the lowest season, usually April is the lowest driest season in Korea, and then in April the relative humidity in soil dropped till almost to the 30%, which means also water is so little for seeds for germination. So once the rehabilitation was conducted, and then we could change the soil condition dramatically within a couple of years. So for the trees and vegetation to survive in those areas that disturbed, and then it is really important to change the conditions to facilitate the vegetation coming back. So, and then we could learn that trees cover those things. So we could control, forest restoration work contribute to climate regulation, and then air, and then soil. And also the second is private forest restoration really helps soil improvement. This is the soil properties changes in Korea. And then in the left side is the soil data in 1981. And then this middle part is the lower part is the soil data in 2003. So they are about 20-year difference. And then here, this is right. I just put the numbers in 2021 soil data. And then in 1981, Korea we planted trees a lot since 1960s. But usually in 1970s, Korea really had big national planting activities happened. So in 1981, it was still like Korea was planting trees. And especially if you see the organic matter, the soil surveyed was all over the country. And then organic matter is, except the lowest one, it was less than, usually it's 30%. So we had really soil was infertile, and then we did not have that soil organic matter in most of forests. While after 20 years, it was improved, so we could see over 3% of the soil organic matter. So the change is soil organic matter. And then if you see the CEC and the cation exchange capacity, and also changed dramatically between like 1980 to 2000. And then in 2020, Korea Forest Research Institute reported that the current condition of Korean forest soil is not that bad anymore. So organic matter average organic matter becomes 4.6%, and then even cation exchange capacity improved a lot. So thanks to national tree planting activities, and then we could witness that soil physical property, soil chemical properties improved a lot but it took about 40 years to get improved soil. So usually we could see soil temperature or those climate factors respond fast. And then soil chemical properties, it takes a couple of decades, but still it's getting improved. And then the biodiversity issues. We planted a lot of trees during 1960s and 70s, and this is statistics of the tree species and number of trees that we planted in 1960s and then 1970s to 1980s. And in 1960s and 70s, because the soil was so poor, and then most of forests were like bare, naked forests. So because the soil was so infertile, so even we could not try the native species so much. So the thing we did was we planted black locust, that is acacia, Robinia pseudoacacia, and then alders, all those species, and then Pinus rigida, the peach pine. So peach pine from the US, and then also black locust from the US. And we planted really a lot of those trees. And then in 1980s and 1990s, because first thing we did was we tried to stop landslides. So we planted trees in the worst spot. And then after like 1 decade, and then we started working for making more forest productive trees with better growth. This is why after 1973 and usually it's 1980s. And then like Korean pine, Pinus koreansis, and also this larix species, larix, that these are the trees that grow to more better soil. So those are the species we tried. So in 1960s and 70s, usually it's Pinus rigida and Robinia pseudoacacia and aloes. Then it's been about 50 to 60 years since we Yes, thank you. Yeah, this is the condition of forest in 1960s. So there is no way we could plant trees on those rocky areas. This is why we try to accelerate soil formation. We just broken the rocks and then tried to plant trees. So this is why we planted Pinus rigida and Robinia pseudoacacia on this land. And then now in Korea, in 50 to 60 years, now in Korea, we still have 1/3 of the forest are planted forest plantations, but it was not for timber production. It was more like for just forest rehabilitation. And then we have a lot of precipitation coming in the summer. That is the time where trees grow. Now most of forests are changed to mixed forest. Like first we planted peach pine, but those quercus species are dominant species most of the country, and then quercus species coming back. So these are, if we see if we visit the peach pine plantations or alders are mostly, they are gone. And then black locusts are also very little remained. And then those native species are coming back with the soil improvement. Yeah, so this is just the typical scenery of Korean forest in the spring. So if you see these cherry trees are planted by the government because Koreans like cherry trees. So they would like to see the cherry tree festival. So local office government planted cherry trees in the areas where the more easy access. And then these broad-leaved species, this is light green, are coming back. So they are naturally invaded, naturally regenerated underneath. And then in the ridges or on the slopes where the soil is still not really good, the pine trees are left. So peach pine or the Korean red pine left on the ridges or somewhat like a dry area. So we could see half of area was changed and succession was going on. So forest restoration works definitely change conditions of temperature, soil moisture content. So the trees, other trees, and more native species can coming back. And then second, it also improved soil chemical quality, soil nutrients. So another native species that require more better soil as can coming back. And the third, biodiversity, because of this broad-leaved species coming back. So now our major disease and then insects to threatening Korean forests are changing. We still pine is the major species, so we have to deal with a lot of pine disease. But still, this disease are more increasing in disease attacking broad-leaved species. And then the next lesson is that I've been to different tropical areas, and they are very different, and their climate was very different. So they have all like deforestation. And then areas where there are no canopy trees or no canopy area, and then because there's no trees on the canopy, so they cannot provide shelter or shading. So the seedlings get drought stress, and also areas in wet season, there is soil erosion occurred. And then seedling mortality or regeneration failure or furloughed. Okay, so this is the area where this is Mediterranean area, but this is the virgin forest in Indonesia, and it's beautiful. But once this is Mongolia, and then when I visited Mongolia, and I was very fascinated because this is so hilly, so people don't have to go, we can use machine to plant trees. So I thought maybe we can just introduce the planting machine and then just use the one day with just plant. And then I was wondering why they don't plant trees there. And then I learned that in spring, their temperature difference was so high. So the temperature in the spring, it goes to like 10 Celsius degree, but at night, it drops down to minus 10 Celsius degree. So if you plant trees in the spring, and then they got froze to die in the night. So Mongolia area, those area has to deal with all the big daily temperature difference. And once there used to be forest, beautiful pine forest, but once they are destroyed, because once there are pine forest, and then the mature trees protect seedlings. There are shading, and also there are like the extremes of temperature can be protected by the upper canopy. But once big trees are gone, and then it's so difficult to restore forest. So I just show you the temperature regime in Korea and precipitation. And Korea always boasts that we were successful in forest restoration because climate helped Korea. We have high precipitation in the summer during the tree growing season. But in Korea, we don't have to deal with this high fluctuation of temperature in the growing season. But there are other areas they have to deal with the daily big changes in temperature. It's okay to temperature changes, seasonal temperature changes, but daily differences are really difficult for forest restoration. But they planted trees. And then because of those things, they have to drain. And then also they have to do have to water the area. So it was a couple of years to make the trees grow. But once they were successful in make trees grow about like 2 meters height, and then the branches overlapped. So this is also the same spot in Mongolia. So this is plantation So they were successful on forest restoration and hope it could last forever. So I witnessed that active action really needed in these areas where they are susceptible in climate air. And then this is the picture of Timor-Leste, the country that is next to Indonesia. So it belongs to the tropical forest, and then they have dry season and a wet season. But it has very long history of forest exploitation. And because this 90% of the land is mountainous region, and it has a wet season, and they get a lot of high precipitation. And tree grows there throughout the year because it's tropics. So they have to do photosynthesis. But during the dry season, they don't get much precipitation. While wet season, once forest is destroyed, and then they have high precipitation during the wet season, and the soil erosion occurred really high. Okay, and then all the soil coming down to the cropland. And then they just swept out all the agriculture during the cropland season. And because there's no trees on the mountains, so during the wet season, they cannot store water. So they used to have 2 times of agricultural activities in like 20 years ago. But now they do agriculture only once a year. And that's next to Indonesia. And now Indonesia is doing 3 times per year for agriculture, and they're pushing them to 4 times per year. But Timor-Leste, they had to reduce it only 1. And then even only in the wet season, they do the agriculture. But during the wet season, soil comes down. And the thing is that also this is the island. So the water, forests store the water, and then they just put the water coming down, but because no water coming in dry season, so the salt water from the sea coming in. So now they have to also give up their cropland because the salt water coming into this area because most of the area is mountains in the center, and then cropland is all like along the coast. So agriculture activities are keep decreasing. And also mangroves. Mangroves has different issues because of the climate change. Elevated sea level is a big problem for them. Of course, the shrimp culture and those things quite destroyed mangrove forests, but now it's climate change. So they are also suffering. Okay, so I learned that Korea was lucky, but other regions, I thought maybe tropical countries, it's hot, they don't have to prepare winter. So why they cannot grow trees? Forest restoration would not be that difficult like countries in the temperate region because we experience winter. But they have different issues and more difficult issues. And once in the tropical dry forest, like Dr. Kaori says, once it's destroyed, and it's much more difficult to protect seedlings and new vegetation coming in because there's no shelter. And that's really important because trees have to grow throughout the year. They don't have dormant period. While trees in the temperate region, they get dormant. They have lucky to have winter. So also forest destruction in tropical forests can be really problem. And then the last thing I talk is that I learned that large-scale reforestation has to be conducted by the government. It's not can be done by the private sector because forest restoration gives all the benefit like good air, clean water, water amount, everything. But that people share. It doesn't go to just the private people. So people know that it's important, but people really need some financial return. But forest restoration doesn't give financial return for a specific person. It's public goods. So this is why public goods. And then also Korea, it takes us 50 years or 60 years to have those water amount by this time. Maybe it took about 20 years to have water. So Korea has semiconductor industry. It's big. I really hope it to continue, but I don't know. But anyway, semiconductor industry needs a lot of water. And Korea in 1960s, no trees on the mountain. There would be no way that we could try the industry, those like semiconductors. So, but I don't think any private company would think of that. And also because it's all like forest races conducted by the village people. So we need also policy and implementation and social regulation is required. So when I visited Timor-Leste, and then the government is poor, so they cannot provide even like any financial return. So there is like United Nations people, and then JICA, and then COICA all coming in, USAID, all different organizations coming in. So that is the area with all different sorts of aid organizations. But because we all they are very experienced in public health or the education and all the medical care, but they are not have, they did not have many experience in forest restoration and forest rehabilitation. Even there are not many people in foresters or forest-related people working in those international organizations. So because the world is really need forest restoration to provide food, water, and those things that are indirectly related to forest, but it's extremely directly related to forest. So because of this fact, it's government and international organizations' job to restore forest. Then I think the foresters and then people who major in forest has to be more actively involved in those activities, especially in the international activities. Okay, so I will start my talk now. Thank you so much.
Yes.
Nice presentation. Actually, I visited your country so many times. It's very similar like China. Back to 1960, you have invested lots of money replanting. Most of those forests, I think, just a single species you planted, particularly the conifer trees.
Right.
Now, how did you do with those forests which already already established, but is in terms of biodiversity is relatively low in facing the climate change, the challenges. You need to so-called some of the silviculture approach on increasing the biodiversity and adapting to the increasing drought.
Thank you very much for the question, because I realized I forgot to point out one important issue in my talk. In Korea, yes, we planted single species. And then the species we used are, I said, pitch pine. And then black locust, it's not conifer, but still, it's a pitch pine. But thankfully, they are pioneer species. So I learned that it's really important to try pioneer species if you don't know the area really well, because pitch pine grows only till 20 to 30 years in Korea. And black locust, they also live only 20 to 30 years in Korea. So now they are kind of succeeded by the native species. So I think it's really important to try the pioneer species. I mean, if you try, it depends on the condition of the site. But if the site had some native species remained, then soil was okay. So it's okay. And it's okay to try the native species. But in Korea, we planted a lot of sugi or hinoki, the Japanese trees. But they have shade tolerance. And then once you establish a sugi plantation, and sugi stays there forever. But while pitch pine, they are gone because they are shade intolerant. So it's really, so I think we can have two different tracks. But once it's a really extreme infertile site, maybe you can just try. It could be okay to have like introduced species as long as shade intolerant and the pioneer species, because they can provide space for native species later when the environment gets better. This is why we have now our native species are coming back. But we are still struggling. Now it's climate change. And then conifers in Korea are not doing well. So we try to change it to more like native broad-leaved species. But the thing is that this Quercus needs more deep soil. And soil depth in Korea has not been fully recovered yet, and it will take another couple of 100 years. But still, if they need at least a 10 to 20 centimeter soil to coming back. So it's slowly coming back on the hills and skirts of the mountains. And so this is why we also try to, we planted conifers because of the root structure and then shallow soil depth. But still, government try to expand. But we are still in the testing period to try different broad-leaved species that could survive in the different soil conditions. Thank you.
Thank you, Dr. Park. I'm going to introduce one more speaker before lunchtime. And that's Dr. Chen Lei, who's from the nearby Institute of Botany and Chinese Academy of Sciences. And I've known him for many years since he was a very young fellow, but now he's the director of the C4 Bio, the network of large plots in China. He studied here in Beijing forestry originally, and then did postdocs again in the United States. He was where I think first met him. He was starting postdocs in the United States, and now he's back here. So he's homegrown, but went off to study for a while. And so he's been involved with large plots that I've been involved with for many years. So I'm very happy to have him speak about work he's done in China.
Okay. Thanks, Rick, the introduction. My name is Lei Chen from Institute of Botany, Chinese Academy of Sciences. Before my talk, I would like to express my deepest gratitude to the organizer, Rick Condit, for his invitation. And I'm interested in forest community ecology, especially in understanding how species can coexist in forest dynamic plots. And 20 years ago, I just got my bachelor degree in this university. So this is a really great honor to have the chance to stand here. Today, I will deliver a talk about how mycorrhizal fungi and species interaction influences diversity and species abundance in a M-dominant tropical rainforest. Okay, conspecific negative density dependence, also known as CNDD, is a permanent hypothesis in explaining species coexistence, especially in hyperdiverse tropical rainforest. CNDD occurs when the focal tree, you can see the focal tree here, when it is surrounded by all species, that conspecifics. So their performance will be reduced. In this case, rare species will be favored over common species because rare species have limited number of individuals, so they have less chance to encounter their conspecific neighbors. So rare species will be favored over common species via the conspecific negative density dependence. And when the species abundance increase in the community, the species per capita growth rate will decrease. So this is also called the community compensatory traits, which is proposed by Joseph Cornell in 1984. Okay, there are many mechanisms can driven the conspecific negative density dependence. I have mentioned two mechanisms here. The first one is about the niche differentiation among species. Conspecific species, they have nearly similar niche, so we will compete strains compete intense with each other so that a conspecific negative density can arise. Another mechanism is a very famous hypothesis, also known as the Jianxin-Cornell hypothesis. It is a special nature enemy related hypothesis. In a community, when species is common, So they have a higher chance to suffer from the local pathogen exposure. So if the pathogens are specialized, so this species, the common species will suffer stronger conspecific negative density dependence. Despite we have a lot of knowledge about the pathogens on species coexistence, however, recent studies have showed that mycorrhizal fungi have also played an important role in species interaction and structure the community. For example, the laboratory just in the new pathologist tensor review, they just showed that negative density dependence can not only can be determined by the pathogens, determined by the effect of pathogens, but if the tree species interact with the mycorrhizal fungi, and the mycorrhizal fungi will protect the species from the negative effect of pathogens. So just show example here. You can see that graph here. When the species have associated with mycorrhizal fungi, the mycorrhizal fungi will construct the common mycorrhizal network and transfer the hypertrophic species to get more resources from the soil or have higher stress tolerance. So these species will be escape from the effect of CNDD, so that you will find in some tropical or temperate forest, you will find especially for that ECM associated species, that will result in the ECM tree species monodominance forest. So this is very common, especially in temperate forest and in some cases in tropical rainforest. In forest communities, most tree species can form mycorrhizal association with soil mycorrhizal fungi. And we will find that most species can form mycorrhizal fungi with the M fungi. You can see about more than 70% species can form M associations, and about 2% of species form association with the actomycorrhizal fungi, that means the ECM fungi. So this mycorrhizal fungi can benefit the plant species survival or growth directly by increase the resource acquisition and stress tolerance, or indirectly by ordinary species interactions. In a large-scale study in temperate forest, they just found that the mycorrhizal fungi can influence the species plant-soil feedback. That is similar to the conspecific negative density dependence. They showed that the M species suffer significant negative plant-soil feedback. You just can see this part. And for the ECM tree species, they just exhibit the positive plant-soil feedback. However, in our recent study in a subtropical forest, we also find this kind of pattern, but we focus on the conspecific negative density dependence. We just found the M species suffer stronger conspecific negative density dependence, but that not was hold for the ECM species. And we combine the seedling dynamic data and the high throughput sequencing data. And we found variation among species in the CNDD was explained by the fungal accumulation rate. If the species have a higher accumulation rate of pathogens, they will suffer stronger conspecific negative density dependence. Otherwise, if they have a high rate of accumulation ECM fungi, like here, they will have experience less conspecific negative density dependence. Okay. Mycorrhizal fungi can benefit plants in variable ways, and they have a lot of benefit. And in a recent studies by Professor Hotaduso, they provide a review paper in Science in 2020, and they highlighted the importance of actomycorrhizal fungi in structures of plant population and community biology. However, as we know, compared to the well-recognized law of M fungi in tropical rainforest, because most tropical rainforests are dominated by M tree species. So we often assume the M fungi play an important role in tropical region. but we still don't know how ectomicrofungi can structure the tropical rainforest. So this question is very important because it is provide an alternative explanation for the latitudinal gradient of CNDD and the diversity patterns, because strongest CNDD in tropical region may be due to the lower abundance of ECM fungal or ECM trees inside the forest. So this is very important. Another knowledge gap is that although we know mycorrhizal fungi play important role in community assembly, but we still don't know in which ways mycorrhizal fungi will influence the community structure and species coexistence. Based on the modern coexistence theories, they have identified several mechanisms or processes that mycorrhizal fungi will influence species coexistence. For example, the mycorrhizal fungi can interact with the pathogens and the plant soil feedbacks and then influence conspecific negative density dependence. That means mycorrhizal fungi can influence species coexistence through via altering the stabilizing mechanisms. Another way mycorrhizal fungi can influence species coexistence is that mycorrhizal fungi can influence the mycorrhizal dependence of the species. And different species may have different benefits from the mycorrhizal fungi. And if common species are favored by certain mycorrhizal fungi, so the fitness difference among species will increase. Otherwise, if the rare species was favored by the mycorrhizal fungi, so the fitness difference among species will decrease, so that diversity can be maintained. In this case, based on this knowledge gap, we just propose 3 questions just want to answer. The first question is whether ECM fungi contribute to plant dynamics in tropical forest. Yeah, we know ECM fungi contribute to plant dynamics in temperate forest and in subtropical forest, but we don't know how it works in tropical forest because most tropical forests are dominated by AM tree species. There's a very, very limited number of ECM tree species there. The second question we want to answer is the pathways by which mycorrhizal fungi, for example, the AM or ECM fungi affect species coexistence. The third question is we also want to know how mycorrhizal fungi contribute to forest biodiversity. We know there are many rare species, just Professor Liu mentioned that. Rare species is very important. There are so many rare species in tropical forest. We just want to know why there are so many rare species. This is a key question in community ecology. To address these questions, we based on the forest approach of Chinese Forest Biodiversity Monitoring Network, we just select a tropical rainforest. This is the Nabanhe tropical rainforest, just here in Yunnan province. The climate here is typical tropical monsoon climate with distinct dry and wet seasons. And we have a 20-hectare tropical forest land plot there. And this is census just like follows the forest geo protocol. All trees with DBH larger than 1 centimeter was measured, mapped, identified to species and recensed every 5 years, just like that. In addition to the tree census, we also monitoring seedling dynamics in this plot. We just erected 159 2 by 2 meters seedling plot in this forest land plot. So we monitor the survival and the recruitment of these seedlings. And you can also see in this forest, most of these species are AM species. And the AM species account for over 95% of individuals. And the ECM tree is very limited in their forest. So we just want to, this is a typical forest that is suitable to answer our questions. We just want to know how ECM fungi will work in this typical tropical rainforest. Okay, and we in 2000, this seedling plot was erected in 2018, and in 2019 we just sample the surface soil in each seedling plot. Yeah, and we then use the high throughput sequencing to sequence the soil fungal community. And based on the fungal tree database, we just assign these fungal OTUs to different functional groups. And these groups include saprotrophs, plant pathogens, and the ECM fungi, and the M fungi, and others. And we use data to link this soil fungal community data to our seedling dynamic data. Okay, how we can test how soil fungi influence the community? We just test how the seedling survival was determined by the effect of soil fungi, neighborhood, and their interactions in this way. So here is the focal tree, and we calculate its neighbors. We calculate two types of neighbors. The first type of neighbor is the seedling neighbor. The seedling neighbor is just, we calculate the conspecific seedling density and the heterospecific seedling density. That means, the density means the number of conspecific or heterospecific seedlings within each seedling plot. And we further divided the heterospecific seedling density based on their mycorrhiza type. So we further calculated another two heterospecific density, so co-mycorrhiza seedling density and the heteromycorrhiza seedling density. And we also calculated the tree neighbors, because the tree neighbors is difficult to estimate, because the neighborhood effect will be biased by the size of the trees and the distance of the trees, and also the nonlinear response of the neighborhood on seedling survival. So we follow this static equation to calculate the neighborhood variables for the tree neighbors. Similar to the seedlings, just you can see the conspecific tree density and the heterospecific tree density, and also co-mycorrhiza tree density and the heteromycorrhiza tree density, just like that. Okay, this is our result. As we all know, in AM-dominated forests, ECM fungi may be limited, and many people just doubted their role, the ECM role in tropical rainforest. But based on our high-throughput sequencing, we found some very interesting results. You will find here the total OTU is fungal species, just like that. We find there's only 5%, about 5%. Of OTUs belong to ECM fungi. This is very low compared to temperate and subtropical forest, because for example, in our subtropical forest study, we just found about 20% of OTUs belong to the ECM fungi, but here is about 5%. This is very easy to understanding because there's limited number of tree hosts in this plot. So this is very easy to answer because ECM fungi have a high specialized to their host species. But we find this here when we account to the relative abundance of ECM fungi. And we found it's accounted for about 90% of ECM fungal abundance. This is really amazing. Yeah. This number is very comparable to that of subtropical forest. So based on this study, based on this result, we just know maybe ECM fungi play an important role in structures the tropical rainforest. And we just do a correlation analysis. We also find that the distinct effect of abiotic and biotic factors were more obvious when considering the ECM fungal relative abundance. Here is the ECM fungal relative abundance. You will find the ECM fungal relative abundance was negatively related to plant pathogen abundance, and positively related to ECM tree density and ECM seedling densities. And another interesting result, we just found a negative relationship between relative abundance of ECM fungi and the soil nutrient level. So in resource-rich soils, there are less ECM fungi and also more pathogens in that forest. Okay. Okay. And we also found the ECM fungi was widely distributed across this seedling plot. And we use generalized linear mix model. We just found ECM, the relative abundance of the mycorrhiza the fungi have a significant effect on the seedling survival. So in subsequent analysis, we just focus on the effect of relative abundance of soil fungi. There are different pathways mycorrhizal fungi can influence the species coexistence. We propose a testable framework, try to empirically test the pathways, how mycorrhizal fungi will influence the species coexistence. According to the contemporary coexistence theories, mycorrhizal fungi can influence species coexistence. I just mentioned there through the stabilizing process and also the equalizing process. The stabilizing process means that any mechanisms that will limit species, limit conspecifics more than they limit their comparators. So it can be tested by dividing the neighbors into conspecifics and heterospecifics. So CNDD means conspecific negative density dependence. And CMDD means co-mycorrhizal negative density dependence. That means comparative exclusion among co-mycorrhizal tree species. And HMDD means the hetero-mycorrhizal negative density dependence. Okay, how mycorrhizal fungi will influence these processes? By review the papers, we just found mycorrhizal fungi can influence the stabilizing process through 3 mechanisms. The first mechanism is soil nutrient partitioning, because different mycorrhizal fungi may help species to use different kind of resources. So it will increase the stress of conspecific negative density dependence, but decrease or mitigate the heterospecific competition. But if mycorrhizal fungi can also influence stabilizing mechanism by reverse Jansen-Cornell effect, that means mycorrhizal fungi can protect the plant against from the pathogens, the negative effect of pathogens. So there's more mycorrhizal fungi, the CNDD will decrease. And if the mycorrhizal fungi influences coexistence through the nutrient trade via common mycorrhizal network, so it will mitigate the stress of conspecific density dependence and also the competition among co-mycorrhizal species. This is how mycorrhizal fungi will influence the stabilizing processes. But in another way, mycorrhizal fungi can also influence the equalizing processes through altering through the differential mycorrhizal benefit. Just as I mentioned previously, if common species was favored by the mycorrhizal fungi, so the species difference will increase, but rare species was favored by mycorrhizal fungi, so the species difference will decrease, so diversity can be maintained. So this is our hypothesis. We just use different models to test our alternative hypothesis. This is our best model. We just test how different soil fungal group, functional group interact with the different kind of neighborhood variables. And we found ECM fungi have a positive effect on the seedling survival. Yeah, this is common, and we all know that because it can protect species against from the negative effect of pathogens. This is very easy to understand. Another reason we can get this kind of conclusion is that most of the seedlings are M species. So they cannot facilitate, ECM fungi cannot facilitate survival of M seedlings through the common mycorrhizal network. So the only way is that it can protect the seedlings against the pathogens. Another interesting result, we just found the positive interaction between ECM fungi and the conspecific seedling density. We will find the increasing ECM fungal abundance was associated with a decrease in the stress of CNDD. With such a negative effect being observed only when ECM fungal abundance was low. You can see this line. At a very low ECM density level, seedling survival was negatively affected by the conspecific seedling density. But with the increasing ECM abundance, relative abundance, this kind of pattern disappears. Yeah. Further, we just focused on the conspecific and the heterospecifics. Yeah, in our previous neighborhood calculation, we also divided the heterospecifics into co-mycorrhiza and the heteromycorrhiza neighbors. we tested how the soil fungi can influence the corn mycorrhiza and heter mycorrhiza interactions. And we also found there are some very interesting results. We will find that ECM fungi enhanced the positive corn mycorrhiza density dependence. That means increasing ECM fungal abundance enhanced the positive corn mycorrhiza density dependence, with such effect being observed when ECM fungal abundance was medium or high. You can see this 3 line. The red line is in high ECM fungal level. You will find the seedling survival was positively affected by the corn mycorrhiza tree density. That means if the seedlings are surrounded by higher density of corn mycorrhiza trees, yes, their survival will increase, especially at high ECM fungi level. Yeah, this pattern is very, very interesting. Yeah, because most of the seedlings are M seedlings, just as I mentioned. So they cannot benefit from the corn mycorrhiza network through the ECM fungi. So one possible explanation here is that the common mycorrhiza neighbors mean 2 things. The first thing is they can construct the common mycorrhiza network. Another thing is they will share pathogens. So if there are more ECM fungi here, so pathogen, the negative effect of pathogen will decrease. And the positive effect of common mycorrhiza network will increase. So we will find this kind of positive interaction. So this is a very interesting result. And we also test, based on our framework, we also test how mycorrhiza fungi will influence species coexistence by altering equalizing processes. We test how mycorrhiza fungi influence the seedling survival. And we find no significant variation among species in mycorrhiza effect on survival. Based on the likelihood ratio test, we found the p-values was larger than 0.05. So it's not significant. That means mycorrhiza, we don't have, that does not mean mycorrhiza fungi cannot influence species coexistence through the equalizing process. Just means that we don't have evidence to support this hypothesis. Maybe we need to do more studies to test whether this, because we cannot measure the mycorrhiza dependence directly. We only measure the mycorrhiza benefit here. So this is a shortcoming of this study. Okay. What was? Ooh. Okay. Previous analysis, we just test how ECM fungi influence the conspecific neighbors and also heterospecific neighbors. Yeah, but we all know species the stabilizing effect of neighborhood interaction not only determined by the conspecific neighbors or the heterospecific neighbors. It is determined by the net effect of conspecific neighbor effect and the heter neighbor effect. That means we need to calculate the stabilizing CNDD. Stabilizing CNDD means the difference between the CNDD and the HDD. And because a stabilizing effect only arises when a negative effect of neighboring conspecifics that exceeds the effect of heterous neighbors. The stabilizing CNDD is very easy to calculate. We can calculate based on this first equation. Yeah, we just the stabilizing CNDD equals D equals A1 minus A2, the coefficient. So A1 is the coefficient for conspecific neighbor, and A2 is if coefficient for the heterospecific neighbors. But here we have come across a problem. We wanted to test how the mycorrhiza fungi or soil fungi will influence the D here. Based on this equation, we cannot directly test the fungi can influence the moderate effect of D. So we use a reparameterized model proposed by Huffman in 2024. We just transform this model through these processes, and then we get the last equation 2. And based in the equation 2, we can calculate the coefficient for the conspecific neighbors directly. But when we adjust it for the total density, because the conspecific density add heterogeneous density, that means the total density. So in this case, the A1 max is equal to A1 minus A2. So here we just the A1 max is directly related to the DE values. That means stabilizing CNDD. So in this case, we have an easy method to test how soil fungi will mediate the stabilizing CNDD. So this is our result. And we found seedling, the stabilizing effect, CNDD effect was differ between the seedling neighbors and the tree neighbors. You will find the stabilizing CNDD is negative for seedling neighbors, but this was not significant. But for the tree neighbors effect, stabilizing tree neighbor effect, we find is significantly negative. And another interesting result, we just found 2 interactions, especially for that of ECM fungi and the conspecific seedling neighbors, and also conspecific tree neighbors. This was significant, but the direction was opposite. How to interpret this result? I just wanted to use 2 fingers to illustrate this result. Okay, when the ECM fungi, the relative abundance of ECM fungi increase, so the stabilizing CNDD for the seedling neighbor will decrease. And the negative value for the stabilizing CNDD, that means it will promote species coexistence. When it was larger than 0, it will not. It will destabilizing species coexistence. So for seedling neighbors, you will find with the increasing ECM fungal relative abundance, the stabilizing CNDD was decrease here. But for the effect of adult trees, you find the opposite patterns. You will find the stabilizing CNDD for the tree neighbors was increase with the increasing ECM fungal abundance. So our result found, this result is very interesting because previously we just think that ECM fungi maybe mitigate the CNDD and cannot promote species coexistence. Yeah, this is a common aspect. But in our study, we just found the dual role of ECM fungi on diversity maintenance. For the seedling neighbors, the presence of ECM fungi will mitigate the stress of conspecific density dependence. And then the stabilizing CNDD will decrease. But for the stabilizing CNDD for the adult tree, I just mentioned, we found significant interaction between ECM fungi and the co-mycorrhiza neighbors. So the ECM fungi will enhance the positive heterospace effect. And so that we will find, because the stabilizing CNDD equals to the coefficient for conspecific neighbors minus heterospecific neighbors. So if the coefficient for heterospecific neighbors is positive, so we will have a larger negative value for stabilizing CNDD. So we've just found the dual role of ECM fungi on diversity maintenance based on our study. To make a summary, we just found, I just identified the pathways by which mycorrhiza fungi will affect species coexistence. We just identified maybe 3 ways. The first way is through the reverse Johnson-Cornell effect. That's due to the law of ECM fungi. ECM fungi will mitigate the stress of CNDD, the first pathway. And the second pathway is the presence of ECM fungi will enhance the common mycorrhiza positive network, positive interaction for AM species. And we also infer, this process is we make inference, yeah, because common mycorrhiza AM neighbors have a positive effect on the seedling survival. So we think the nutrient trade via common micro network maybe also works in this community. And the most important is that we just find the dual roles of ECM fungi on species coexistence. The last question we just wanted to answer is how mycorrhizal fungi contribute to plant diversity and why there are so many rare species. The question is very important because many studies have showed that rare species is very important for the ecosystem service in forest communities. And based on the BCI forest dynamic data, and they just found in BCI there's over 300 species. And the most abundant 9 species account about half of the individuals in that plot. You can see there are many rare species, more than 161 species only accounted for 1.6% individuals in the plot. So many rare species there. Stephen Hubble and Liza Kamita, they have do seedling synthesis in PCI forest dynamic plot. And they test how neighborhood interactions influence the seedling survival. They just find the conspecific neighbor effect was varied significantly across the species. We are found here. But the heterospace neighbors was not significant across species. And when they linked conspecific negative density dependence to the species abundance, they found a very interesting pattern. Yeah, this is a positive relationship. They just found rare species suffer stronger negative density dependence. But as I mentioned, CNDD provide a stabilizing effect only when common species are favored over rare species. But here, rare species are not favored, but common species are favored here. Yeah, so it is very interesting to see this pattern. And this pattern also verified by another field and greenhouse experiment in BCI. Yeah, they just do study and found that, for example, these 6 species, they have different streams of plant soil feedback. And when they rate the plant soil, streams of plant soil feedback to the species abundance in the BCI forest, and they just found this kind of positive relationship. And the rare species, most rare species experience the strongest negative plant soil feedback. And they do the analysis, they attribute this pattern to different mechanisms. They categorize the mechanisms, for example, the insect herbivorous, and also foliar pathogens and others, soil biota. And you will find that the insect herbivore and the foliar pathogens play a limited role to drive this pattern. And most of this pattern was driven by soil biota. So they think that this pattern just generated by a hypothesis named enemy susceptibility hypothesis, because different species because rare species may be more susceptibility to the pathogens, so especially the soil biota. In a recent published book by Stephen Hubble, they just proposed this hypothesis in his book. And they think that this hypothesis asserts that rare species owe their rarity to being more susceptible to root and heart rot fungal pathogens, or share more of their pathogens with other host species than to common species. This is very easy to understand. They just provide new explanations for why there are so many species in tropical rainforest. Just because rare species are rare, because they are more sensitive to pathogens. So this is a new hypothesis. We just wanted to test this hypothesis to test whether this phenomenon was also can be observed in our tropical rainforest, in the Naban River tropical rainforest. Okay, we use a multi-level hierarchical Bayesian model when we test how the constant neighborhood effect varied across species and how the streets related to species abundance. So this is our findings. X-axis is the species abundance, the base area, and the y-axis is the conspecific seedling neighbors effect. And we find the conspecific seedling neighbor effect was very significant across species. And we also linked it to species abundance. We find a similar pattern in BCI. and the other tropical rainforest. So this pattern is very common now in tropical rainforest. But whether this pattern was driven by pathogen, we just do another analysis. We just divided the neighbors, divided the focal species into different categories based on their rarity. And the rare species, we just follow Stephen Harper's criteria. So rare species means if the species with less than 20 individuals per hectare in forest land plot. And the common species is just with larger than 20 individuals per hectare. And this is our finding. Okay, you will find for common species, ECM fungi have a positive effect on the common species seedling survival, but no significant effect on the rare species. And we also find the significant interaction between conspecific seedling neighbors and ECM fungal abundance, the positive interactions. But here, the rare species suffer stronger conspecific negative density dependence here. So, but we also analyze the effect of pathogens, but we don't find any difference between the rare and the common species. That means the rare and common species difference in negative density dependence maybe not due to the pathogens, but just due to the effect of ECM fungi. Because compared to rare species, common species get more benefit from the presence of ECM fungi. So we just provide alternative hypothesis, but we needed to do more studies, we needed to have more evidence to know whether this pattern was really is real. Yeah, this is the key. And we just found the rare species was not favored over common species. Yeah, so we just think that the presence of mycorrhizal fungi will destabilizing the species coexistence, and consequently we predict the presence of mycorrhizal fungi will decrease the seedling diversity. Yeah, we just wanted to provide indirect evidence. We just use the SAM models and to test how mycorrhizal fungi influence the 2-year changes in seedling richness. For each 2x2 meter seedling plot, we can calculate the diversity change in each seedling plot. And this is our result. We found the relative abundance of ECM fungi significantly reduced the seedling diversity. And another way is ECM fungi was significantly positively related to pathogen channel diversity and still negative related to the seedling richness. And we also find the presence of M fungal diversity also have a negative effect on the seedling diversity change. And this pattern is also very easy to understand, because when we divided the species in rare and common, and we also found the common mycorrhizal seedling density and the seedling survival have a positive effect when M fungal diversity was higher. That means M fungal will also favor common species over rare species. So in this case, rare species will get lost in the forest community. Okay, let's get a summary. So the first question we can answer is yes, ECM fungi also contribute to plant dynamics in tropical rainforest. It's very important roles. And the second, we just found two major processes. So first one is moderate the stress of CNDD and the positive common mycorrhizal feedbacks. And the last question is we found the common species are favored over rare species as a result of mycorrhizal fungi, and consequently maybe impair the diversity of seedling assemblages. Okay, I want to thank my collaborators and the funding agency for their support of these studies. And that's all, thank you.
It's really a fascinating talk and forces me to think about a lot. Although everything you told me doesn't support rare species, they should all be going extinct. Which, I think, well, we can talk about that. I think we can talk about that over lunch. Let's do that, because it's a very difficult set of analyses. And I'm glad you're doing it, that someone's doing it. So we will break for lunch. Thank you very much, Dr. Chen. we have in the period here, there's some posters out in the hall that we would like you to have a look at and read, and you could try to find speakers if you'd like and ask some questions, and try to find poster presenters for questions. And we have this lunchtime that gives you a chance to talk and discuss among yourselves. We will be back here at 1340, correct? All right, thank you very much for the morning session. To all the speakers. Okay, that's what I mentioned. Yeah, please read the posters during the break if you can.
各位老师，我们可以凭我们的参徽证直接去就